In this video, we're going to be talking about 1 through 3 through 1.6. It's a lot. So it's about 100 slides, and it's all about biological macromolecules. So we're going to be diving into the intro of it, properties, structure, and finishing up with nucleic acids. So it is a pretty important and hefty unit. So what I would do is split it up so that you're not just so focused all at once, and pause it, take notes, look things up that you don't understand, and really engage yourself in the learning of it. So 1.3 dives into hydrolysis and dehydration synthesis. 1.3 continues on to talk about polymers, monomers of the macromolecules, and then it just keeps going through 1.4, 1.5, and 1.6. So what I want you to do is take this essential knowledge, and at the end of it, figure out what you know about the essential knowledge and what you still need to continue learning. So fill in those gaps that you have. And one example of that is after you finish it, okay, you read DNA and RNA molecules have structural similarities and differences related to their function. So think about what you learned and think about how what you learned relates to that essential knowledge. So the purpose of doing this before our actual class that covers it is you can come in with questions, and you already have some background knowledge when we dive deeper into class. And another important part is that in class we're mainly focusing on skills. So one of the skills is describing a biological concept based off of the model that's represented visually. So essentially what I want you to do is have the knowledge so that we can start applying that knowledge in class to the skills. I feel like that's a more beneficial time and a time that's more applicable to what questions you'll be asked in the AP test. Okay, so to start, molecules of life. There are four main classes of biological macromolecules: carbohydrates, lipids, proteins, nucleic acids. If I say carbs, I mean carbohydrates. So macromolecules, you can break it up. Break up that word. Macro means large, molecule. It's a large molecule made up of a bunch of atoms. So it's usually huge, and they're consisted of polymers, which consist of monomers. So what I would relate this to is Legos. So think of one Lego piece like a monomer, and it's a molecule that can react to form long chains of repeating subunits. So just like how one Lego can attach to another Lego, one monomer can continue attaching to become a polymer. A polymer is a long molecule consisting of identical building blocks of monomers, and they are linked by covalent bonds. So in a Lego, you have the Lego piece, and you have that little circle at the top of it that links it to another Lego. Think of it like that's a monomer. The circle in the insert is the covalent bond, and then when you attach a bunch of those together, you form a polymer. And they are linked by covalent bonds, but each of the covalent bonds have different names. So I do want you to know the different names of each macromolecule and of the bond specifically. But they all are covalent. Okay, so the way that they are formed is through a reaction called a dehydration reaction. And a more specific word, so think of it like dehydration reaction is a larger umbrella term, but more specifically, the type of the reaction that occurs is a condensation reaction. That's why it's red. It is a type of condensation reaction called a dehydration reaction. I think I got it flipped in my explanation just now. So it is a condensation reaction, and the specific name for it is dehydration reaction. So if you want to think about the word dehydration, think about it like you're dehydrated, you're thirsty, you need water, like you don't have water. In a dehydration synthesis, what happens is you're removing the water from the monomers being formed, or the monomers attaching to each other. So here we have two monomers. We have one monomer here. Oh, there's a pointer. Cool. We have one monomer here, and if you look at it, there's an OH. There's a hydroxyl group attached to it. Monomer 2 also has a hydroxyl group attached to it. Right now, the oxygen is bonded to something, which is bonded to a hydrogen, and it has 4 extra electrons on the outside. So 1, 2, 3, 4. And then it's sharing 1 from that hydrogen, sharing 1 from this of the covalent bond. Now, in a dehydration reaction, what happens is one monomer has a hydroxyl cleaved out. So that's cut. That's why it's blue. You cleave the hydroxyl from that monomer. And in the second monomer, you have a hydroxyl group. But instead of the entire group being cleaved out, you just have the hydrogen that is removed. So if you remove the hydroxyl from this group, you now have an oxygen that is in need of another bond. And then in monomer 2, you have a hydrogen that was just removed conveniently. And now this bond can be fulfilled by that extra hydrogen. Now you have 1 oxygen and 2 hydrogens. That forms H2O. So you're removing H2O from the reaction. And as a result, now you have an oxygen that is in need of 1 bond. And there's a monomer that now also needs a bond. So that oxygen right there can covalently link together the 2 monomers. So now the monomers are linked by a covalent bond. That is a dehydration reaction. And the reason why it's called a dehydration reaction is because of the removal of that hydrogen, or that water molecule. And this reaction requires energy. So it is one that requires energy, and as a result, that means that it needs an enzyme or some type of protein that can continue to enter the situation to speed up the chemical reaction to allow it to occur. And enzymes decrease the activation energy required for a reaction to occur, which allows this to happen. So energy is required for bonds to form, for a polymer to be formed. And the enzyme assists in that energy requirement. So this is how long chains of polymers is created. Hydrolysis is the breakdown of a polymer. So hydrolysis is the reaction that is used to break the bonds. So think about like hydro is water, lysis is to break. So you're using water instead of taking water, you're using a water molecule to break the bonds. So think of it like the inverse of what just happened. If you remove a water and that forms a bond, you add a water and that breaks a bond. So you're using water to break the bond by essentially doing the opposite. So if you break the covalent bond between that oxygen right here, now that oxygen needs another bond for it to be stable. That hydrogen from the H2O will come and fulfill that. So now you have the hydroxyl for monomer 2, and now monomer 1 is locking an entire OH group. Luckily, from that water molecule, you still have an OH group left, and that attaches to monomer 1, and now they are 2 separate monomers. Okay, so hydrolysis in our body occurs pretty often. So when we eat, we need to break down that organic material. And we have different places in our body that continues to break it down. And let's say you eat like a carb, and that's a polymer. You need to break that down into its individual monomers so that the body can use it. So hydrolysis is the reaction that occurs. And in the same way, when we break it down, we use it in different areas. And as a result, how it gets used is, or how like those monomers continue to get applied into our bodies is through dehydration reactions. So we're breaking down the what we eat, the organic material that we eat, so that we can have the individual subunits which are carried throughout our body. And then when our body needs to make a polymer, it uses a dehydration reaction to form a polymer. So it's a cycle that occurs. And then before I continue any further, I'm going to pull up some notes on the side just to make sure that I don't miss anything. Okay, cool. So this slide right here is showing essentially the digestive tract. So as we eat, our food enters the mouth, and the digestion occurs through 2 different ways: mechanical digestion, which is like chewing and swallowing, And then we also have chemical digestion of carbs and fats. And as it continues down, there are other places where chemical digestion occurs, and it's of different things. So you can see like mechanical and chemical digestion continues to occur until it gets either utilized in our body, and then the extra waste is released through our anal sphincter. But one thing that you may be familiar with is your saliva. So amylase is essentially the enzyme that uses that allows and facilitates for the hydrolysis of starches into smaller carbohydrates. So it takes the purpose of your saliva is actually to break down different foods as a means of chemical digestion. And this is another picture that kind of shows the exact same thing, but you can see that like hydrochloric acid is also another one inside our stomach that is used to hydrolyze proteins. So it denatures it and hydrolyzes it. So we have different types of enzymes or acids in our body that help facilitate or specifically hydrolyze the different macromolecules that we consume in organic matter. Okay, so let's dive into our first type of macromolecule in more depth. This is a carbohydrate, and it's a biomolecule with mainly carbons, hydrogens, and oxygens. They include like things like sugars, starches, fibers, and when you think of carbohydrates, one thing that most people talk about is sugars. So there's a lot of carbohydrates that we may consume. So think about when we eat bread, when we donuts, pasta, all of that carbohydrates. And essentially, like if you think about the different types of food inside our network, a lot of times like our diet consists of a lot of carbohydrates because they're very filling, but at the same time it's also something that we have to regulate a lot. So the monomer of a carbohydrate, so remember that individual Lego or that individual subunit of a carbohydrate is a monosaccharide. So mono 1, and then saccharide or is sugars, so it's simple sugars. Polymer is the disaccharides, double sugars, or like polysaccharides for polymers. And one question that I want you to continue asking is what reaction joins this to form a disaccharide? So what reaction is facilitating the joining of two different monomers to form a polymer? And this is also how I want you to study. So as you're going through this, continue asking yourself questions like carbohydrates are biomolecules that consist of primarily what? So it's almost like a Cornell method of note taking, but as you're going through a lecture. So in college, one thing that helped was actually just printing out the slides. And instead of having to write down everything on the slides, what I could do is just write down questions that I would use to ask myself later on. So I would take this slide and I would be like, okay, what are the monomers of carbohydrates? What is the polymer? What reaction joins monomers to form polymers? And you're beginning to build that knowledge through that. So it's the macromolecule specifically of carbohydrates is a polysaccharide. Polymers have many sugar building blocks. This slide continues to break down what I just stated. Mono, single, saccharide, sugar. The molecular formula, it's very similar to CH2O. So carbons, hydrogens, oxygens. So if you see a lot of carbon, hydrogens, and oxygens together, you can kind of deduce that that would be a carb. Don't worry about the chemical formula. I would say focus more on being able to recognize that this is what is an indicator for a carb. And if you're confused about like what goes into a carbohydrates in regards to like the different atoms or elements, think about glucose. So you know that sugar is a carb. Glucose is a huge part of different processes. Again, I want you to ask questions as you're going through this. Where do we see glucose? So this one should bring you back to the idea that glucose is present in photosynthesis, cellular respiration, chemical formula C6H12O6, carbon, hydrogens, oxygen. So think about glucose if you're ever confused. I don't think I'll ever ask you a question like CHO is indicators of what. But If it does, just think about glucose. It's a sugar, and you can see that it has carbon, hydrogen, oxygen. And it has a carbonyl group and a hydroxyl group. So these are the functional groups that kind of are specific to carbohydrates, or not specific, but more indicators of carbohydrates. So a carbonyl group, not carboxyl, carbonyl group and a hydroxyl group. And you can also recognize a sugar by if it ends in an ose, so like sucrose, glucose, sugars, fructose. So if it has an ose, just know that it's within the groups of sugars. And the location of the carbonyl plays a role in what kind of sugar it is. So there's two different types. So there's the aldehyde sugars and there's also the ketone sugars. And these are also isomers of each other. So glucose and fructose actually have very similar, they have the exact same molecular makeup. They have carbon, hydrogen, oxygens of the same amount. But what makes them different is where the carbonyl group is located. So if the carbonyl is at the end of the chain of carbons, then it's essentially a glucose or an aldehyde sugar. But a ketone sugar has the carbonyl group inside the skeleton. I think next slide does not have it, but if you look at this right here, it actually has the functional group at, give me one sec. I just want to make sure before I say that. Okay, cool. So it has the carbonyl functional group at the end. So you can ask yourself like what type of sugar this would be. So if you look at the carbonyl group right here, it's located at the end of the carbon chain. So therefore, the type of sugar that this would be is a aldehyde sugar. So monosaccharides are the monomers of carbohydrates, are big parts of nutrients for our cells. So think about cellular respiration. Glucose plays a huge role in cellular respiration. So we need the nutrients from the monosaccharides to carry out essential functions. But they're also important in the synthesis of other molecules. So amino acids and fatty acids have makeups that we take from the breakdown of carbohydrates. So if they're not used for different things like making amino acids or making fatty acids, then they're eventually just created for mono or polysaccharides, or they're used as mono or polysaccharides. So mono or polysaccharides. So they have a lot of importance in our bodies, not just as carbohydrates, but carbohydrates help influence nucleic acids. They help influence lipids. And you'll see this trend throughout the entire lesson in that different things, like let's say nucleic acids who have DNA, are responsible for coding or having the information that's required to make proteins and what those proteins will look like. So all of these different macromolecules influence each other and they play a big role in each other. And the type of covalent bond that carbohydrates form is called a glycosidic linkage. So remember when I said that covalent bonds form all of these different macromolecules, but they all have different names? This is the first one. So a glycosidic linkage is a covalent bond that is formed through a dehydration reaction. All of these are formed through dehydration reactions. But they all have different names and they all are covalent bonds. Okay, so one common disaccharide is sucrose. Sucrose is table sugar. So just like how we have NaCl, sodium chloride, table salt, the sucrose is the generic table sugar that we see. And there's two monomers that make up sucrose, glucose and fructose. And then if you if you change that fructose, and instead of having a glycosidic linkage between these two, you have a glycosidic linkage between glucose and galactose, you now make lactose. So different monomer makeups or different monomer combinations influence what type of carbohydrate or what type of disaccharide occurs. So these are two that you see very commonly, sucrose and lactose. So again, as you're studying, ask yourself questions like, uh, where do we see lactose present in milk? What is the purpose of lactose in our bodies? And this is in a different language, but we can kind of see, we can still understand it. So if you look at it right here, we have the two hydroxyl groups. We have hydroxyl group here, hydroxyl group here. And remember, one whole hydroxyl group is cleaved off. And in the other one, the hydrogen from the oxygen of the hydroxyl group is cleaved off. And as a result, you have a covalent bond between that oxygen and the other monomer. And you have galactose mixing with glucose through a dehydration synthesis. And you can see the bonds right here. This is a glycosidic linkage, which is a covalent bond to form lactose. And if you look at the opposite arrow, so you're adding H2O in the opposite arrow. And if you add H2O, you know that's a hydrolysis reaction. And when you add water, that breaks bonds. So here you can go back and forth. So to go forward, dehydration, you're taking out H2O. To go backwards, hydrolysis, you're adding H2O and you're breaking the bonds. Okay, so the structure of monomers determine whether it's an aldose or a keose. So think about the different types of sugars, aldehyde sugar or the ketone sugar. So one of them is the carbonyl group at the end, the aldose, and the carbonyl group located within the carbon chain is a ketose. So aldoses and ketoses react differently and bond differently due to the location of the carbonyl groups. This leads to variation in the disaccharide or more complex carbs that lead to variation in its function. So function is determined by the form of the monomer. Okay, polysaccharides. Polysaccharides are the macromolecules of carbohydrates. They are polymers with hundreds to thousands of monosaccharides joined by glycosidic linkages. They are used for sugar, in the cells, storage, and other functions. Okay, so one carbohydrate that's super important is starch. And you may have heard of starch before, but have not known what starch is. But it's a storage polysaccharide of plants, and it's made up of a bunch of glucose monomers. So if you see a bunch of glucose monomers together, it's a storage polysaccharide or a storage carb, and it's joined by 1,4 linkages. So there's a bunch of carbons inside the rings of, like, let's say glucose, and in order to like specify which carbon it is, they number the carbons. So you'll see like 1, 2, 3, 4 for the carbon numbering. And in the carbon numbering, it helps you identify like, okay, this carbon is bonding to this carbon. It helps you keep it consistent. So these are just like names for these specific carbons, like identification markers. And starch is stored in plants in granules called plastids. So chloroplast is actually one of those storage granules. So on this slide, it kind of zooms into it. You can see that like you have different things like the cell wall, and if you really dig deeper, you have the chloroplast right here. Inside the chloroplast, you have dicolloid membranes, and inside that, you have starch grains. So starch is stored inside things like the chloroplast, and chloroplasts are key because they allow for like capturing of the energy of the sun. So in animals, I have to use the arrow key. We actually have it, we have storage molecules, but it's called glycogen. So essentially that is like the starch of humans or animals, vertebrates. And humans store this inside their muscles and their liver. So inside your muscles and your liver is where you would find our storage. So some questions to ask yourselves. How can the sugar within the starch carbohydrate be utilized? So what reaction must occur in order for the starch to be utilized? So right now it's being stored. Like, okay, if you're storing something, you need it later. You have to pull it out. So what reaction is allowing for that starch to be used? And the second question is, what do glycogen and starch break down to? So what do they break down to? So what are the monomers of the polysaccharide? And there's two main ones. So the first one is starch, found in plants, stored in plastids, including chloroplasts. Glycogen is found in animals, and that one's stored in muscles and liver cells, or our liver. Okay, and to answer your questions, the first one, reaction must occur, hydrolysis. And it breaks down into glucose, because that is the specific monomer or monosaccharide that it breaks down to. If you just say monosaccharide, you would be technically incorrect because it's looking for something more specific. So there are different types of monosaccharides. Okay, structural polysaccharides. So one use of a carbohydrate is storage. The other one is structure. So we use this to build strong materials. Cellulose is a big one. It makes up the cell wall in plant cells. So plant cells, if you remember, differ from human cells in that we don't have a cell wall, but plants do. And that's made from cellulose, which is made up of mainly glucose, but there's a difference in the position of the glycosidic linkage. So glycogen and starch. So what we have is we have different shapes. We have if it's inside the alpha position. So if you look at the alpha position, the number of this is why I said the number of the carbon is important. So if you look at the number of the carbon, you have a 1 carbon right here, and if you look at this one, you have a 1, 2, 3, 4. So that's the 4th carbon. So 1, 4 carbon. And if you look at the location of the oxygen, they're different in terms of the 3-dimensional shape. So one of them is bonded to the alpha position, which creates kind of like this shape. And then another one's a beta position, which is kind of like a sheet. And cellulose would be into the beta position. Glycogen and starch is the alpha position. So exact same thing, like glucose. They're both glucoses, but they're just bonded to different positions, which change what the carbohydrate is used for. So this is really just a slide emphasizing the idea that the bonding location also influences the purpose of the purpose of the carbohydrate. And going back to that picture right here, you have the cell wall. Now we know that's made of mainly glucose, but the glycosidic linkage is different. It's on the beta position, which allows it now to be used as cellulose to make up this really strong cell wall found in plant cells. So here's a question. Can the enzymes that hydrolyze starch also hydrolyze cellulose? So I want you to think about that question, pause the video, write down a predictor, or write down what you predict from it. And then you would find that the answer, and look it up too. And you would find that the answer is nope. So the enzymes that hydrolyze starch cannot hydrolyze cellulose. And this is actually important when you think about, like we consume plants, but what happens to that cellulose? So we have enzymes that hydrolyze different things, but not starch or cellulose. So we don't have the enzymes to hydrolyze starch, and cellulose just kind of passes through, and we can't hydrolyze these bonds. Hydrolyze means to break it down, so it kind of stays into its polysaccharide or disaccharide form. And because we can't break it down, we can't use it inside our bodies. So what that's called is an insoluble fiber. So if you heard about what fiber is, like if you ever hear a doctor say like you need more fiber, or or somebody saying like you need more fiber, you usually need more something to help your digestive tract like pass better. And one common one is a fiber 1 bar. And what they do is, because we can't digest it, it lines the tracks of our digestive system, and it allows food to continue passing through it easier. So if you think about if you're struggling with like constipation, one treatment for that would be more fiber inside your diet. And just to test it out, I went on the Fiber One website and I looked at the ingredients, and check it out. We got some cellulose scum mixed in here. So we have different things, and just by understanding like the purpose of a structural polysaccharide, and also understanding that we can't break down like cellulose, it helps to understand how a fiber 1 bar is a fiber 1 bar and how it aids in our digestive system. Okay, so here's another carbohydrate, structural polysaccharide is called chitin. So chitin is used by arthropods, which is like spiders, crustaceans. So if you see like different crustaceans with like hard shells, or exoskeletons where there's like an outside skeleton that surrounds a soft inside, that is carbohydrates. So it is cellulose, similar to it, but it's different in that it has a nitrogen appendage. So if you look at this nitrogen right here, this is what cellulose lacks. So you add in this nitrogen appendage, and now you have an even stronger structural element that makes an exoskeleton. And it's made of something called chitin, which is a structural polysaccharide. So carbohydrates have so many uses in so many different animals, plants, crustaceans. So carbohydrates are used in various different means and methods, just based off of like what you want to build. So complex carbohydrates comprise sugar monomers whose structures determine the properties and functions of the molecules. So the structure, the function of the molecule is determined by the properties of the monomer. So how the monomer is, what type of bonds that it can interact with, they all make up complex carbohydrates that we can kind of tell the properties of based off of the monomers. Lipids. So lipids are the second class that we're going to talk about of macromolecules. They are hydrophobic molecules. So what that means is they essentially have no affinity to water. Affinity is like attraction. They don't want water. If you break down the words hydrophobic, hydro, water, phobic, like if you have a phobia, that's like a fear or something. So you have a fear of water. Just think of it like they don't want to be around water. And it's a direct result of their structure. So the reason why is because they're mainly hydrocarbons. So if you look at these chains right here, so this is essentially a hydrocarbon, and if you look at it, that point right there is a carbon. So there's a carbon there, and they don't show the the hydrogens bonding to it, but there are hydrogen bonding to that. So there's a carbon, 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 carbon. So a bunch of carbons together, and then hydrogens are attached to that carbon. And the three most common types are fats, phospholipids, and steroids. So we're going to be talking about each one of these. And here's a picture of some lipids. Okay, so they are not polymers, but they're huge. So if you think about polymers, they have monomers, but lipids don't necessarily stem from like an individual subunit, but instead they're just hydrocarbons, a bunch of carbons. But they can vary in different sizes. And if you think about this picture right here, it kind of reaffirms what I was just saying about how they are hydrophobic. So if you put oil and you put water together, you can even try this at home if you have some like kitchen oil and some water. Put them together, shake up the bottle, and it's not going to mix. They're going to separate because it's there's no like electrostatic interaction that would occur between the water and the lipid. So think of it like there's no desire for water to bond with the lipid, and there's no desire for the lipid to want to be around water. Like they're all like okay with just being themselves and without meshing. So I'm trying to find my notes one more time. I close the tab. Okay, so one question that you want to ask yourselves is, why do fats separate from water? So I want you to think beyond the fact that they're hydrophobic. I just said that like 10 times. but why are they hydrophobic? So think about what we learned from like past lectures. Why are they hydrophobic? And you should have paused this video, and you should have thought of it, wrote down an answer, or looked it up. But one reason why they're hydrophobic is because they're non-polar. So remember, polar is like a difference in charge, or having some electrons move over to the oxygen because it's more, it has a higher electronegativity and wants to attract those electrons. That doesn't happen in a lipid. So that doesn't happen in carbons and hydrogens. As a result, there's no electrostatic interaction between the polar water. Remember, water is polar because of that, the electrostatic difference between oxygen and hydrogen. So the electrons move towards the oxygen, giving it that negative charge. But there's like, it's non-polar. Non-polar don't interact with polar. So that's why they're separate. And then what reaction connects the bonds that form fats? The answer should be dehydration reaction, which is a covalent bond. So this is important though. So it's not important for you to understand like the chemical structure, but it's important for you to know that a fat is made from a glycerol, which is an alcohol with 3 carbons and a fatty acid. So if you don't know what a glycerol is, I would look it up right now. So this is important. Understanding that fat is stemming from a glycerol and a fatty acid. Fats can be liquid or solid, and the type of linkage that it is an ester linkage. So that's the name of it. Yeah, an ester linkage is the name of the covalent bonds. So just like how carbohydrates had a specific name for the bond, lipids' specific name for the bond is ester linkage. And when you have 3 fatty acid molecules joining with a glycerol, so glycerol is almost like the base of it, and then you have 3 fatty acids, you get a triacylglycerol. So the importance of this is essentially just understanding that 1, ester linkage is the covalent bond, and 2, fats stem from 3 fatty acid molecules with a glycerol, triacylglycerol. You may see this a lot, especially if you enter the medical profession. Okay, so there are 2 types of fats that you can kind of describe. You may have heard of this before, but saturated and unsaturated fat. And like specifically more in terms of like, oh, you want to eat this type of fat more. So let's talk about the differences between the 2. So a saturated fat is a fat with no double bonds. So fatty acids differ in terms of, do they have double bonds, or do they not have double bonds? So a saturated fatty acid has no double bonds, and because it has no double bond, it has extra bonds that it needs to like fulfill in order for the carbon to be stable. Remember, carbon forms 4 bonds. If there are no double bonds and the carbon's attached to another carbon, okay, so now it still needs 2 more. So now 2 hydrogens attached to it. So that's a full carbon. And if you, in the long term, if you look at like the chain of it, which we will see soon, let me see, in a few slides, there's a lot of hydrogens in a saturated fat. Now, a unsaturated fat has double bonds. And because it has double bonds, think about it this way. So you have a carbon attached to a carbon. So that's 1 bond, but because it's double bond, it takes up 2 of them. And remember, carbon can form 4. As a result, it has 2 hydrogens. I think I made a mistake with the other one. Here's what we'll do. We'll go over here. I don't have a, dang it. I don't have a drawing tool on my laptop because it's not a touchscreen, but no worries. Here's what we do. We'll drag this one over. So this right here is a saturated fatty acid. So you see the carbon chain right here, and then you have the glycerol. So if you look at this right here, you have carbon, there's no double bonds, so it can just attach to 2 hydrogens. But here you have a double bond. Because it takes up two bonds, it can only attach to one more hydrogen because the other one's a carbon. So it can only have one hydrogen here. Whereas saturated fatty acid doesn't have a double bond, so it's saturated with hydrogens. So if you want to think of an easy way to remember, saturated means full, like you're saturated. Nobody says that, but saturated means like full. So it's full of hydrogens. Unsaturated, it's not full of hydrogens. So, and if you look at this picture right here, you see that there's this kink. That's because double bonds are very stiff. Single bonds can rotate around and they can move around. They're more, they're less stiff, but double bonds are very stiff, so it forms a kink inside the chain, and that's one unique feature of the unsaturated fat. And at room temperature, it's a liquid. The reason why is because it can't pack together as tightly because there's a double bond. Think of it like this. You take a metal straw, right? And then at room temperature, it wants to pack tightly, but you can't push a metal straw together. Okay, this right here is a metal straw, pretend. I can't push it in to form, to pack it tightly. So it stays a liquid because it's more spread out. And if you go back to chemistry, you would know that everything is tighter inside a solid. So this right here would stay a liquid because it's stiff, restricted, and it can't pack together. But at room temperature for a saturated fat, it can pack tightly together. So that's like butter. And then olive oil would be the unsaturated. So butter would be the saturated, olive oil would be the unsaturated. So saturated, unsaturated. I was supposed to draw it here, but I realized that I didn't have my iPad near me, so I can't draw that there. But just use that picture. Just go back to the video when I just dragged in that image. That's what I was going to put here. Okay, so a diet in saturated fat contributes to high risk of cardiovascular disease. Arteriosclerosis is essentially occurring in patients with higher diets of saturated fats. The reason why is because it develops plaque inside the blood vessels and it restricts the blood flow. So if you're going to eat fats, try to limit the amount of fats that you eat, but also eat unsaturated, because when it is used to replace saturated fats, it does contribute to a decreased risk of cardiovascular disease. And we can dive deeper into that image right here. So what happens is the unsaturated fats form plaques and clog the arteries, which restricts ultimately the blood flow. So normally it doesn't have any plaques right here, so it can continue to move. The blood can continue to flow, but with that plaque, it hardens and it's right here. So another way to think about it is we know that saturated fats are more fluid and they can also pack tightly together because there are only single bonds. They're saturated in hydrogen. So in the same way, it can pack tightly into plaque and contribute to the congestion of our arteries and create abnormal blood flow. So another function for lipids is, I don't think we talked about it yet, but one function for lipids is energy storage. So we store fats, humans store fats or animals too, in something called adipose cells. So adipose cells, they shrink when fats are removed, and they also swell when it's full of fat. So think of it very variable depending on how much fat is stored. And it's actually okay, so let's talk about adipose tissues first before I move on. So adipose tissues are consisted of these cells, and they cushion the organs like the kidneys and insulate the body. So you have a bunch of adipose cells which ultimately form adipose tissues. Adipose tissues, fat tissues, cushion organs, and they also insulate the body. So it's good. I mean it's good to a certain degree. There's a healthy amount of adipose cells and tissues within the body that is required, and they do provide warmth. And if you want to think about lipids as a function for energy storage, just think about let me find my slide. Okay, Anyway, so just think about it, like we need energy, and we need to store it somewhere. So it is stored inside like reserves, and we want to use those reserves when we can. So like let's say like our body is like really fighting something and we can't take in as much nutrients, then it starts pulling from those long-term reserves. Okay, phospholipids, the second important lipid. So instead of 3 fatty acids, remember triacylglycerol has 3 fatty acids attached to a glycerol through ester linkages. This one has 2. And then the extra spot is filled with the hydroxyl group. So if you remember from the first, I think, lecture, we talked about functional groups, and we talked about how functional groups, the properties of functional groups help you understand and predict like properties of different things. They also help predict the properties of macromolecules. So you have a hydroxyl group right here with a phosphate group. So both hydroxyl and phosphate, you want to think about the functions and characteristics of it. So I ask a question here, what are the functions of hydroxyl and phosphate, or the characteristics of it? And the answer is they're polar, that's number one. And remember, polar interacts with water. Electrostatically, they interact with water. And secondly, phosphate is charged, so it's negatively charged. So now you have a polar, you have a charge attached to a fat. So it's kind of like interesting, because you have like one aspect of the phospholipid that does not want to interact with the water, and you have another part of the phospholipid that does want to interact with water. And you get a hydrophobic tail with a hydrophilic head. So hydrophilic means like affinity to water. They want to interact with water. And a hydrophobic tail don't want to interact with water. So what you get is you actually get something like really cool bilayers. So what a bilayer is like 2 layers. So you have one layer of phospholipid right here and another layer of phospholipid right here, where the hydrophobic tails go on the inside and they what's it called? They interact with each other to form a wall of hydrophilic heads on the outside, prevents water from interacting with it, and it's happy. The hydrophobic tail is happy. The hydrophilic head is also happy because now they're interacting with water. And the key term for that is amphipathic. So make sure you look that up. It's red, so it's important. Okay, steroids. Third one that we're going to talk about. Carbon backbone with 4 rings. So 1, 2, 3, 4 rings, and the carbon backbone's right here. So, or you just see it throughout actually. So they're not just steroids that you are thinking like inject steroids into you so that you can get buff. It's also includes things like cholesterol, and that's something that we all have. So steroids goes beyond just like injected steroids, and they are also like steroids that are used to treat asthma. Like I believe one steroid is prednisone. So prednisone is used as like a bronchodilator. So it dilates and allows you to breathe better. It's also used as a treatment for laryngitis, I believe, and also asthma. So that is a steroid, and cholesterol is also a steroid. And this right here is an animal membrane, and you can see if you look inside it that there's cholesterol. You can actually see the rings too. Pretty cool. So there's 4 rings right here in this animation or this drawing, and those 4 rings, those are cholesterol. They embed itself within the phospholipid bilayer, and because they embed themselves inside the bilayer, one question that you can ask yourself is, what property do they have? Like, are they polar? Are they non-polar? You can probably predict that from just like looking at it, but just by the way that it's like embedded into the non-polar hydrophobic tail, kind of gives you an indication of like it doesn't have anything polar within it, which is why this part of the cholesterol is inside there. And it's in the animal cell membranes, and it's a precursor to other steroids. What that means is cholesterol is used to make other steroids. And many hormones, like sex hormones, are produced from cholesterol. So it's good. You need a certain extent of cholesterol. But too much of it, once again, cardiovascular disease. So you want to like moderate cholesterol levels. And I believe we'll be talking about like LDL and HDL, like low density lipoproteins later. But for now, we're going to just move on to the basics of cholesterol. So cholesterol in the membrane is good because it does these 4 things. Strengthens the membrane, So by embedding cholesterol in there, strengthens the membrane, decreases its permeability. So I posed that question earlier. What characteristics of it? It's nonpolar. So it decreases the permeability of the bilayer, phospholipid bilayer, and it reduces packing in the membrane at low temperatures. So remember, at low temperatures they want to come together. So think about like butter at low temperature, it turns into a solid. So it packs together. But if you have cholesterol in there, think of it like you're trying to squeeze like something soft together, but you put something hard in the middle, and now it's harder to squeeze it. So it acts like to prevent packing of it. But also it raises the melting point at high temperatures. So it does two things. At low temperatures reduces packing, at high temperature reduces melting. These are important, these four. All right, summary. Lipids are nonpolar macromolecules. The differences in saturation determine the structure and function of lipids. So saturation, think about like unsaturated and saturated. Phospholipids contain polar regions that interact with each other like polar molecules, like water, and with nonpolar regions that are hydrophobic. So it has that tail that's hydrophobic and also the head that is hydrophilic. Okay, next is proteins. Proteins are pretty important. So simply enough, 50% of dry mass of cells, it makes up like half of a cell. It's important. And one of the most important types is an enzyme. So enzymes are used as catalysts. Catalysts which decrease the activation energy required to start a reaction. So enzymes like really allow for a lot of things to occur. Remember in the type of reaction that forms the covalent bonds that bonds all of these things. So it goes through a dehydration reaction, and it needs an enzyme to do that because it needs energy. So enzymes help play a role to even form the bonds that form macromolecules. They are not digested after use. What that means is these proteins can be reused over and over. So proteins are really important, and they're also reusable, which is awesome. And there are many different types of proteins, and they all perform different functions. We'll be talking about like what influences those functions afterwards too. So these are 1, 2, 3, 4, 5, 6, 7 different types of examples of protein types. And it's from Khan Academy. You can always click on these. Remember, you can click on these to go to the resource that I got it from. And doesn't want to load anymore. So okay, here we go. I get these resources. What I do is I take the slides from both the textbook and other resources that can help you out. So for example, if the book has something but I think it needs to go into greater depth or needs a better way of explaining it, these resources help. So make sure to click on it because it describes so much of what I'm talking about. Okay, so proteins, you have to know the different rules of it, but you don't have to know all of them. Just know that they have different rules. Okay, the monomer for a protein is an amino acid. I abbreviate that AA. The polymer for it is a polypeptide. So a bunch of amino acids make up a polypeptide. And we'll be talking about amino acids later, but they're influenced by nucleic acids, DNA to RNA to protein. So a protein is one or more polypeptides folded and coiled into different conformations. So a protein is when you have a polymer formed from a bunch of amino acids and you start folding and you start coiling it into different things. This is important. An amino acid has an amino group, NH2, carboxyl group, COOH, and hydrogen atom and one R group. So it has 4 different things. An amino group, carboxyl group, hydrogen atom, and one R group. This is important. Also, the R group is variable. So the R, it's kind of like something goes inside the R. It's like X in like math. So the R group is variable. And then the type of R group determines the structure and function of that protein. So remember how we were talking about functional groups and the type of functional group and the aspect of the functional group, the property of the functional group determines like how that is going to create like interactions, whether it's polar, non-polar, things like that. The R group is the same way. The R group is ultimately what determines the structure and the function of that protein. And there are 20 different types of amino acids that we know. Each amino acid has different properties. So they are positive, like some of them are charged, some of them are not charged. This one has a hydrophobic side chain. This one has a polar uncharged side chain. So there's so many different types of amino acids, and the combination of amino acids create different functions. So amino acids interact differently based off of their properties. Amino acids are linked through what reaction? Dehydration. So think about like the dehydration reaction leads to the type of reaction that's formed. Remember, it removes an H2O, and because of that, it leaves that oxygen that allows it to covalently bond, and then boom, they get a bond. And the type of bond for a protein is called a peptide bond. And if you do this over and over, you create a polypeptide. And there's many different types of amino acid formations that could occur. Amino acids have unique properties which influence the function of the polypeptide. So once again, this is kind of just like me reading through the slide. It's a slide where there's not much other than like the bullets I mentioned. And then we dive deeper into here. So Sanger sequencing. Sanger sequencing, Frederick Sanger found a way to use a protein digesting enzyme. So what that does is it hydrolyzes, cleaves the polypeptide into its separate like amino acids. And with the amino acids, we can take the amino acids and we can put those together. We can kind of like piece together what the amino acid is, and we can determine the sequence of the amino acids that make up that protein. Now the important part of this is, let's say that you have something like insulin, which is needed inside our body to regulate our blood sugar levels. By understanding the amino acid sequence in it, what we are able to do is we are able to take that amino acid sequence and create synthetic versions of what we just sequenced. So this was key in the discovery of the sequence of amino acids, which now allows us to create insulin, and insulin is used as like a mediator or treatment for diabetes to maintain like blood sugar. So that's awesome, like being able to understand the amino acid sequence. It kind of like tells a story of, it's like a blueprint of how to create that protein. And there's a video here that will help you out in understanding that. It is required. Okay, protein conformation and function. So a polypeptide chain does not make a functional protein. What makes a functional protein is how it interacts with each other, or essentially how it folds. So the type of folding is what makes a protein function. So it's almost like unfolded is kind of like the inactive state, and then once you fold it correctly, it becomes an active state. An amino acid sequence determines what the fold will be like. So the reason why that is because let's say that we have like a polar charged and a non-polar. They're not going to want to interact with each other. So the type of amino acid, the property of the amino acid determines how they will interact. Think about like when you put oil and water together, they don't want to interact. But when you put like a phospholipid and you have the hydrophilic head, it wants to interact with the polar water. Same thing here. So in order for it to interact, it needs amino acids that want it to interact. So the properties of the amino acid influence how it's going to fold. It also determines what it can bind to and recognize. So, for example, an antibody is a protein that binds to a foreign substance that invades the body. So we have different antibodies inside our body, and right now with COVID, like we're testing for COVID antibodies, right? If you have the antibody for COVID, what that does is it binds to foreign substances like COVID viruses. It removes that from the body. It signals for it to, like the antibody binds to COVID viruses and signals for its destruction. So if you have the antibody like for something, that's why we're trying to identify COVID antibodies. An enzyme is a protein that binds to specific substrates. So Think about like a lock and a key. They're very specific. So the type of folding of the protein is kind of like determining what will fit in there. And receptor proteins are unique because it's influenced by shape. So like the receptor could be a triangle, and it only binds to triangle things. Or it could be like a circle, and it only binds to circle things. And there's 4 conformation levels. So we're going to be talking about that very soon, like right now. Okay, so there's 4 structures. This is like a basic slide. I have a more specific slide that we're going to get into for each one. But a primary protein structure is just a chain of a polypeptide. It's just an amino acid chain. Then you have a secondary protein structure. There's 2 types. There's an alpha helix, and there's also the beta pleated sheet. We'll be talking about that too in more depth. Tertiary protein structure is when that one polypeptide chain starts folding into different means with combinations of like beta pleated sheets and alpha helices. And then quaternary protein structure is now when you have more than one chain of amino acid interacting with each other. Like another one comes in and now it's interacting with the old one. Now you're getting even more complex folding. This is a required video. I don't know, I think all videos are really required. But this right here is a video that I want you to watch about the protein structures. You can watch it before or after, but it'll help you out. Okay, primary structure. So this is the most basic one. It's just a sequence of polypeptide chains. And insulin is an example of that. It's just a 2 polypeptide chain. And this one here is, I believe, insulin that's not human. It's a cow insulin, but human insulin is very similar. So it's just a chain of polypeptides. And we can determine this sequence right here by Sanger sequencing. So we can determine like the A chain, what it is, the B chain, what it is, and we can actually synthetically replicate it with what we know here. Question: when there's a DNA mutation, how would that affect proteins? So think about that. If there's a DNA mutation, how does that affect proteins? But don't just like give me a basic answer, think about why. So just keep asking yourself, dig deeper into like your answer. Why does it do that? Why does it do this? Okay, if it does this, why does it do that? Okay, secondary structure. This is when you have different types of protein folding. So there's 2 types of folding, the beta pleated sheet and the alpha helix. Simple way of looking at it, alpha helix, think of it like a helix, like DNA double helix. It's spiraling, this one's spiraling. Beta pleated sheet, think of it like a sheet. It's like 2 sheets on top of each other. And they're all held together by hydrogen bonds, which are weak bonds, which allow it to easily coil, uncoil, interact, not interact. The bonds can break really easily. Remember, hydrogen bonds are one of the weak bonds. And the backbone is what interacts. So remember when I said that the R groups are important? It's because the R groups allow for interacting. And also the location of the R group. So if you look at this right here, you can see the R. That's where the R groups are. R groups are variable, which means that things can attach to it and it can differ. But here we can see the location of the R groups. Like for the beta pleated sheet, they're on the inside and the outside. For the alpha helix, they're also on the outsides. And because it's on the outside, it allows it to continue interacting. So think about quaternary structure, how you need one polypeptide chain to interact with another polypeptide chain. So because the R groups are on the outside, it can continue allowing for more interaction. If all the R groups are on the inside, it can't interact. And here's a deeper picture of that. So you can see the R groups are on the outside. It allows it to continue interacting. So this is important that there's 2 types of protein for secondary protein structures, alpha helix and beta pleated sheet. And here are some descriptions of the differences. So the carbonyl of one amino acid is bound to the amino H of another amino acid. So for alpha helix. So if you look at this right here, we have the amino group and we have the carbonyl group, and they're interacting. And they're interacting. So we have the hydrogen of the amino with the oxygen of the carbonyl, and it allows them to interact with the hydrogen bond, which is the 3 dots right here. Oxygen interacting with the hydrogen. oxygen with the hydrogen. Okay, so if you look at that, that's the interaction that's occurring. But if you look at the beta pleated sheet, what happens is the hydrogens form between the carbonyl and the amino acid of the backbone. So if you look at this one, the backbone of the it's the backbone that's interacting with the amino of that right there, which is forming a sheet. So the different types of interaction, this one's coiling, this one is not necessarily coiling, but it's forming like two different sheets almost. Like parallel, they run parallel. And I think I said that too, yep. Okay, so for the beta sheet, they can be parallel, but one important thing is that they can be both parallel and anti-parallel. So anti-parallel means that they're running in opposite directions. Just like DNA, DNA can run 3' to 5', or it does run 3' to 5', and then 5' to 3'. So 3' to 5', 5' to 3'. Yep. So they run opposite directions of each other, like kind of like my hand. So like same direction. I don't think I can do that. This is like the same direction. This right here is the opposite direction. So you look at my fingers, they're opposite directions. This one, they're the same direction. That's anti-parallel. Parallel is they're running the same direction. So they could be either or. Also alpha helix separated by 4 amino acid positions. So the reason why is because they're coiling with each other. So the reason why they're coiling and they have to interact with each fourth one is because as you coil it, you're not going to interact with every single one. You're interacting with every fourth one to create a coil. All right, tertiary structure. So this is a 3D structure of a polypeptide chain. The importance of the R groups can't be understated. The interactions of the R group is what allows for the tertiary structure interaction. And the interactions have different types of bonds because remember the R groups have different properties. So the bonds occur based off of what R group it is. So the bonds that could occur are hydrogen, ionic, dipole-dipole, London dispersion forces. They're all weak bonds. So none of these are covalent bonds except for one, which is disulfide bond, which is links between sulfur and the cysteine. So if you have a cysteine in that group, you can form a covalent bond, but otherwise you're forming, majority of the time, a weak bond. And the properties of the R group is what determines that. You can have polar, nonpolar, hydrophobic, hydrophilic, different properties, different interactions. And here's an example of that. You have a folded protein structure, and if you look super deeply into it, you have an ionic bond in this one, and you have I don't know why I'm pointing like you can see it, but here's my mouse. You have an ionic bond, and you also have a hydrophobic interaction right here. And then hydrophobic interaction is not like a bond within each other. It's not like it's like linking or anything. But remember, in the hydrophobic tail of the phospholipid bilayer, the hydrophobic ends like come back on the inside. Same thing here. It's going to come back on the inside. So it's interacting hydrophobically because it doesn't want to be exposed to the water. So it's like trying to keep itself on the inside. Disulfide linkage. This is the super strong one. The covalent bond between the sulfurs. Disulfide, 2-sulfide linkage. Hydrogen bond. The interaction between a hydrogen bond. And remember, they're really dependent on the type of R group. Different R groups, different interactions. Here's the final one, quaternary structure. So the quaternary structure is essentially when now you don't just have one single polypeptide coming in, now you have another one. So you have at least two chains that are interacting with each other, and they can form really complex structures. So we'll be looking at this picture more deeply. But an example of it is hemoglobin. Holds iron, has 4 subunits, 2 alpha helices and 2 beta sheets interacting with each other. Even more complex, DNA polymerase. We'll be talking about this in like the cell. I think that's like unit 2. And it's the enzyme that synthesizes DNA. Has 10 subunits. So subunits are like chains, so it has like 10 different chains coming to interact. So basically it can be really complex. And here's a deeper dive into it. You have a primary one, single chain. Now you have secondary. It could be pleated, could be alpha helix. And then now they're folding with each other. and then boom, now you have another one come in, and now they're interacting. See the two different colors? Now they're interacting and creating like more complex structures. Now, remember when I asked the question of what happens if there's a DNA issue, and how does that influence the protein? Here's one real-world example of it, sickle cell. So it's a small change in the primary structure affects its conformation. I'm actually going to change how I do this slide. I'm going to do the next slide first. So here we have normal hemoglobin. But now, instead, like we're going to change glutamic acid and valine for valine. So you have an amino acid substitution. Instead of this, you have this. And that creates a sickle cell hemoglobin. So one substitution of the valine, instead of its like traditional one, which is glutamic acid, like totally changes the structure of the hemoglobin. And as a result, the protein that carries the oxygen is now in a sickle like formation. It creates like a sickle cell shape. So you can see like normal cell and a sickle cell. And it leads to like angular cells, which impede blood flow and hold less oxygen. So just one change in amino acid creates like such big implications for the body. And things that can like affect that change, or things that can like throw off protein formation or folding, is pH, salt, concentration, temperature. Any variation in like our natural pH, salt, concentration, or temperature really can change how our proteins fold, which cause issues within our body. It's also why our body is so focused on maintaining homeostasis and like going back to our stable self, because we have to maintain all of this so that we can do things like have naturally folding proteins correctly. Denaturing. I'm just going to say denaturing. So denaturing, a protein loses its native conformation. So denaturing can occur from like heat. So you can denature a protein just by like heating it up. So think about why fevers are so dangerous for the body. You really denature proteins with a fever when your body temperature is higher than normal. And denatured protein is also biologically inactive. So if the folded protein is active, denatured like changes its shape, now it's inactive. And another thing that can change it, chemical agents that disrupt bonding. So for example, like if you transfer a protein from like an aqueous environment to organic solvents, sometimes what happens is the hydrophobic edges are going to shift the outside and it denatures. So I want you to think about why that happens. Another way to think about it is like if we put a protein inside a liquid environment, what's going to be on the outside, like touching the water, and what's going to be on the inside during the folding. So I'm going to go back a few slides. This one right here, hydrophobic interactions. I'm going to bet that this middle part doesn't have water because it's interacting, hydrophobic interaction, but outside here does. Now let's say that we take this out and we put it into an environment with that's not aqueous. The hydrophobic interaction, does that have to occur anymore? The answer would be no. So it doesn't need to occur anymore, so now that protein is going to change its shape. So basically, the specific shape of the protein is really important for its ability to function. The sequence of the amino acid determines how it interacts, because each amino acid has different properties, and the sequence of that is going to change how it folds. Anything that denatures that makes it biologically inactive. So anything that denatures a protein makes it inactive, inability to function. So let's say like this right here is an active protein, an active enzyme. All right, it binds to a substrate. If this protein denatures, a substrate won't fit its like normal thing, right? So let's say like a enzyme has like a triangle active site, and the substrate comes in, and now the protein or the enzyme denatures, and it's no longer a triangle, it's now a circle. It's gonna have issues because now the triangle can't fit inside the circle opening. This right here is just showing normal protein folding, heat, Now it's single polypeptide chain. It's denatured. And something that is also like an essential protein function is a chaperone protein. And a chaperone protein helps in the function or the folding process. This right here is what it looks like. So essentially what a chaperone protein is this right here is like looking at it from the bottom in. So you're looking at it from the bottom in, and the protein like goes inside that hole, and then a cap goes on top of it, and it helps the protein fold by preventing it from getting stuck to itself, or the wrong things interacting with each other. So think about it like this: like you have a long piece of tape and you're trying to like fold it perfectly, but since tape is so sticky, it sticks to itself, and you might not get what you want. Chaperone proteins help in assisting that like stickiness of it. So here's a summary of it. In proteins, specific order of amino acids in the polypeptide primary structure determines the overall shape of the protein. Amino acids have directionality. So it does have directionality with an amino group and a carboxyl group. So it has a carboxyl and an amino terminus. The R group of the amino acid can be characterized by chemical properties. So the R group of the amino acids determine its properties, like is it hydrophobic, philic, ionic, and then the R groups allow for interactions based off of its properties. And next we're going to be moving on to nucleic acids. So make sure that you understood proteins, pause it before moving on if you did not understand it. Okay, we're going to go ahead and finish up with nucleic acids. So there are two types of nucleic acids. We have DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. And the important part of nucleic acids is that they are responsible for carrying all the information that we need to create essential components. So think about proteins, right? Proteins, the information that make proteins have to come from the sequence of amino acids. The sequence from amino acids are determined by codons from DNA. And DNA has to be essentially transcribed to RNA and then translated to a protein eventually. So the information that stems from all of that stems from the nucleic acids. DNA provides directions for its own replication, so it makes itself. So everything, like if you think about everything coming from DNA, what does DNA come from? It comes from itself. So it makes its own replication. And DNA directs RNA synthesis, so DNA to RNA. And we inherit that DNA from our parents. And every chromosome is one long DNA molecule with thousands of genes, and they're kind of just like all together at once. And that's like the blueprint for life, that when it is transcribed and then translated, it's essentially unraveling what the blueprint for life is. Okay, so each gene on the DNA directs messenger RNA, which is mRNA. Sometimes just referred to as RNA, but it's a specific type of RNA. There's also like transfer RNA, tRNA. But the gene on the DNA makes mRNA, messenger RNA. And the messenger RNA interacts with the protein synthesizing machines, which are located at the ribosomes, that direct polypeptide production proteins. So this is RNA, this is DNA. One thing to note, DNA is double helixed, whereas RNA is not a double helix. Oh, also something else to note. Everything's the same except for, so like cytosine's the same, guanine, adenine's the same, and then uracil instead of thymine. This is super important. So the structure of a nucleic acid is actually one of the critical statements or essential knowledge of AP Bio. So it contains a pentose, which is a 5-carbon sugar. So if you look at it right here, we have pentoses. And a phosphate, so pentose right here, and then it's bonded to a phosphate. And then you also have a nitrogen or nitrogenous base, which could be either be adenine, thymine, guanine, or cytosine in DNA, or instead of thymine, you have uracil in RNA. DNA and RNA are different in their function and structure. So one way to look at it is DNA has the deoxyribose pentose, whereas RNA has a ribose. So that's why the D and the R, deoxyribo and ribo. So it's stemming from the type of pentose that they have, or the type of sugar that they have. So make sure that you know these three, what a nucleotide contains. And then if you have just the phosphate and not the phosphate. If you just have the pentose with the nitrogen base, that's actually called a nucleoside. So it's like part of the nucleotide, if that makes sense. And then you add in the phosphate, and now it's a nucleotide. So now it's like a full thing. So 3 components: phosphate, deoxyribose or ribose, so DNA, RNA, and then the base. And an important thing to note about the base, you have 2 different types. You have pyrimidines and you have purines. So pyrimidines are just one ring, and you have a purine, which is 2 rings. So it is important to distinguish the, so I'm looking for my notes once again, scrolling down. Okay, so it's important to distinguish that there are 2 different types of bases. Okay, so pyrimidine is a 6-membered ring of carbon and nitrogen, and cytosine, thymine, and uracil are pyrimidines. Purines are larger 6-membered rings with a 5-membered ring, and that's adenine and guanine. So I'm not gonna like quiz you on is cytosine a pyrimidine or purine. I'm gonna ask you higher level questions in terms of talking about the differences between the structures of it. So you do have to know that which one's which, but understand that the questions are going to be expanded beyond just memorizing which one's which. So learn how to apply that. And if you look at the difference between deoxyribose and ribose, they look super similar. So I mentioned earlier that carbons are numbered in order to understand like the labeling of it, and to understand like, oh, when we're talking about it, we're talking about this specific carbon. So if we start numbering it, we have 1, 2, 3, 4, 5 carbons. The difference, one of the differences is there's a hydroxyl group on here on the ribose, but it's just a hydrogen in the deoxyribose, and that is located on the 2' carbon. On the 3 and the 5' carbon, they both have hydroxyl groups at the end of it, and the 5' carbon gives it directionality. So 3 and 5' carbon. So if you think about DNA, we talk about it in terms of 3' and 5', and 5' and 3'. That is stemming from the pentose. So the 3' carbon ring is where the nucleotides are joined. That's why it's important. So the 3' is what joins the different nucleotides, and they're joined by a covalent bond, and the bond is called a phosphodiester linkage. So just make sure that you understand that, and remember which type of linkage it is. The 5' end is where the phosphate is going to be attached. So 3' end nucleotide, 5' end phosphate, and this creates a backbone of sugar phosphate units. So the sugar and the phosphate units. So you see the backbone right here? That's essentially what's going to happen. So you have a backbone of sugar phosphate, and on the inside you have the nitrogen containing bases. The monomer of a nucleic acid is called a nucleotide. The polymer is called a polynucleotide, formed by phosphodiester linkages. So that's the covalent bond. The directionality of DNA is important to understand. 1, understanding that there is directionality. 2, understanding how that directionality is formed. So what is at the 3' end? What is at the 5' end? That's important. So DNA is unique because of the different sequences. So you have 4 in DNA and you have 4 in RNA. The only difference is the thymine to uracil. But think of all the different combinations that you can have. You can have like A, T, G, C, A, G, C, T. So you have so many different combinations, and as that chain continues to get longer, the more combinations that you can have, and the more variation and the more uniqueness that you can have. And these specify the amino acid sequence. So 3 codons, or 1 codon has 3 nitrogen bases, and the 3 nitrogen bases all code for a specific amino acid. So the variation leads to variation in the amino acid that's produced, and the amino acid that's produced influences the protein structure, and the protein structure and function is influenced by that primary sequence. So really what's happening is that DNA is providing the instruction for what the protein should look like. Nucleic acids, DNA double helix. So DNA has a double helix, which means that it spiraled, and it's 2 of them, double helix. And this was determined by 3 scientists, actually many scientists, but 2 that are recognized for the Nobel Prize, but 1 that needs to be mentioned every single time. So James Watson, Francis Crick won the Nobel Prize for his discovery, but they also have controversy in that there's a lot of talk about how they stole the Nobel Prize from Rosalind Franklin and her discovery and her contribution to it. And she actually died from cancer at the age of 37 and was unable to receive the Nobel Prize as a result of that, because it can only be given to those who are alive. However, she was a huge scientist and researcher in the discovery of the double helix. I don't think that it would have happened without her. So it was discovered, and just to give light on how recent the discovery of the double helix was, James Watson is still alive. I think he's 92. He's still alive. So it's a very recent discovery. So a double helix is 2 polynucleotide strands. So polynucleotide is the polymer from the monomer, which is the nucleotide of a nucleic acid. And you have 2 of them, and they spiral, turns into a helix. And they run in opposite directions. This is called anti-parallel. I did this back inside the protein when we were talking about beta pleated sheets. Anti-parallel. So my fingers on this side is right here, and then my fingers on this side is right here. They run anti-parallel. They're held together by hydrogen bonds and van der Waals forces. So they're very weak bonds. And one thing that I want you to know, or ask yourself, is what is the significance of it being held together by hydrogen bonds? You would want your DNA to be strong, right? But it really isn't. The bond in between it is one of the weakest bonds. So why is that? So research that. The bonding is very specific. So how it specifies correct bonding is the number of hydrogen bonds. So adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3. So you see how there's going to be issues if you try and pair A and G together? One has 2 and one has 3 hydrogen bonds. So that's why they're very specific. A pairs with T, adenine pairs with thymine, guanine pairs with cytosine. They are different in the number of hydrogen bonds. And they form a base pair when they, when I say base pair, I mean A to T, G to C. And if you have an issue, let's say there's substitution for a cytosine to a adenine, now you have 3 hydrogen bonds trying to bond to a 2, and that's not going to work out. So AT, 2 hydrogen bonds, GC, 3. It's important. And this is referred to as complementary base pairing. So we get information from looking at nucleic acids. So we learn a lot by studying nucleic acids. For example, we can study a relationship between different species, different animals, different things. We can determine our relation between other relation. Remember when I was saying the insulin, when we were talking, I think we were talking about proteins, insulin of a cow is very similar to human insulin. We learned that through studying the nucleic acids. Yeah, we could study amino acids too, but where do amino acids come from? Amino acids come from nucleic acids. And they come from the sequence of the DNA, which make up that amino acid. So for example, the sequence of a polypeptide chain for hemoglobin, carries iron, between a human and a gorilla only differs by 1 amino acid. This is why there's so much talk between the relation between a human and gorilla, and also its role in evolution, because they're looking at the number of amino acids that differ. Like humans and monkeys differ by 8. Humans and mice differ by 27. And we understand evolutionary kinships because of this. So we understand how things are related. All right, here's a summary slide. In nucleic acids, biological information is encoded in sequences of nucleotide monomers. Each nucleotide has structural components: a 5-carbon sugar, deoxyribose or ribose, a phosphate, and a nitrogen base. DNA and RNA differ in structure and function. So we're at slide 81 now. So there's a lot of slides for this, but we're wrapping up like the basics of macromolecules. And remember, this isn't like the end all of our information that we need to learn. This is actually just the beginning, because now what I want you to do is take your understanding of what we learned from all these slides in each of the macromolecules, in how they're formed, and go back to the essential knowledge. So go back to the essential knowledge where it says like hydrolysis and dehydration synthesis are used to cleave and form covalent bonds between monomers. Now take that and continue adding on to your knowledge of that. Make sure that you really know that essential knowledge statement. Same thing with this essential knowledge statement. And this one, and this one, and this one. So all of these, go back, continue looking at the essential knowledge statements and seeing how your knowledge fits into that. Ask questions and also use external resources to help you out. For example, come on. Like Khan Academy, make sure that you click on these, you can see it, and it brings you to this. Then you can read even more about it. So what I did is I just took summaries from other sources, which are listed at the bottom, and that will help you understand it. I just collect the evidence, tell you where to get the evidence or the information from, and your job to expand on what you know and what you don't know. Like even videos, make sure that you're watching these videos, because these videos help summarize and add on to your existing knowledge from this lecture. So that about wraps up 1.3 to 1.6. Just make sure that you're tying it back to your essential knowledge statements, and in class you're going to be diving into applications of this through questioning, free response questions, and diving more into explaining it, not just like lecturing on the information during class. So good luck studying.